induced asthma type situation yesterday, so I'm a little hoarse. If I start coughing, it's exercise induced, not because I have COVID before I start. So the paper that I asked you all to read, can you see the PDFs? That you should be looking at PDFs.
Yeah, we can.
Okay. I wanted to make sure I was sharing the correct monitor. So the paper that you were asked to read was looking at this review of the transcription factor network in Mimulus, or monkey flower, which is the organism that I study. And in particular, this reaction diffusion system is the one that I am studying. So the video that Greg sent out last night, evening, hopefully you got a chance to watch it. If you didn't, well, I liked it because it's showing pretty flowers. But it really does a good job motivating why how pollinators interact with flowers and why that's important in terms of the reproduction, because it means that plants don't have to rely on just the wind to carry their pollen. Instead, they can allow pollinators such as bumblebees, or in the case of milkweed, the monarch butterfly, so moths, butterflies, bees, hummingbirds. These are all pollinators that are important to plants in general. But actually, we know that Mimulus as a genera, I think Mimulus is a genera. That might be totally wrong. As a group, rely on a wide variety of pollinators. And one of the things that separates the species of Mimulus is who pollinates them. And the reason that they get pollinated differentially is in large part due to their coloration, which is where this reaction diffusion transcription factor network comes in. So if you envision a transcription factor network as having a promoter region along the piece of DNA, what's happening in our network in particular is that this red tongue is inhibiting the production of the transcription factor MYB, or Negan. Okay, hold on. Who was confused about this whole MYB-Negan RTO interaction? No one? Okay, admit it. Thank you. Let me pull up a slide, because I just realized that I was starting to use this and I don't think that it's necessarily obvious. Okay. So this is the system that we're looking at here. The MYB that we're talking about is this W, usually, okay, sorry, I just got distracted. This RTO is our inhibitor. The reason it inhibits is because the only way for the MYB to bind to the DNA is when it is in this trimer. So the MYB binds with the WD40 and the BHLH. And these are all just little transcription factor protein guys, but they bind to the DNA together. The mechanism that we believe the RTO is inhibiting this by is that the RTO binds with the BHLH. So if you think about a chemical reaction, like Chem 101 type stuff, when this RTO binds with the BHLH is no longer able to bind with the MYB and the WD40, so it becomes like a limiting reactant. And in so doing, we get this situation where the inhibitor or the activator can't function. So if we're talking about the MYB, we might be talking about this piece of the trimer, but more likely we're talking about the trimer in total. This MBW transcription factor is the whole thing. Negan, if you read Negan is the specific transcription factor or the specific form of MYB that we have in Mimulus cutatus. And I forget what it's called in Lewisii. So Negan stands for nectar guide anthocyanin. So we're specifically talking about the MYB protein, which is acting in the nectar guides of the flowers. So when we're talking about the nectar guides, these are the parts that are really very attractive to bumblebees, and that's where we are talking about the bumblebees wanting to land. So these nectar guides are the yellow part of the flower, which is saying, hey, bumblebee, land here, land here. And what we're observing and what we're trying to study is how this speckled red pattern is influencing, is controlled by the transcription factor network in this paper. Now, what I'm studying is more focused on the petals, but for now, we're talking about the nectar guides, which is why we're talking about Negan, because that Negan is the MYB in the nectar guide.
So, okay. Emily, at this point, what would it be helpful? I think it's helpful for people to, if you leave that figure up, you know, so the different panels that are shown here are, this is work by a collaborator of our collaborator, Ariel Cooley, and the wild type flower and nectar guide is shown on in the leftmost column, right? And
yeah,
then,
I was going to come back to this figure after we've talked about the transcription factor network.
So you'll explain the different mutants at that point?
Yes.
Okay. I'll let you.
That was my plan anyway. I'm happy to do it now though.
No, whatever you think works. I just didn't want to, I think a lot of people would not understand what the diversity on this plot meant.
Yeah. So I want to talk about the, so before we go into the diversity of that plot in particular, I wanted to sort of go over this figure in the paper that I actually asked you guys to read. And then I think once we've talked about this figure a little bit more, you'll understand the diversity of that figure a little bit better. So if we look at part A of this figure, this is the way a lot of biologists will represent a reaction diffusion type network or system, where you have the activator on one side and the inhibitor on the other, and you have these arrows. You saw that was actually how I represented it in my prospectus talk as well. And in some ways it's useful, but in a lot of ways it fails. So for one thing, we don't know much about the strength of interaction, nor do we know much about the mechanism of interaction. All we know in this case is that the activator somehow is activating itself and producing more of itself. And it also binds to the DNA to cause the production of more of the inhibitor. Conversely, the inhibitor, when it binds to the promoter region, or, well, it doesn't have to bind to the promoter region, though that's often what people will say, it somehow causes a lack of Negan to be produced. And the piece that's missing is that this RTO we believe also causes a lack of production of itself. So it is also self-inhibiting. And that's the model that we're using in the system that I'm working on right now. Now, these little squiggly arrows are representing the diffusion. So in the paper, they talk about long-range inhibitor and short-range activators. That was a hypothesis that was put forward by Mary Guerrier and Meinhardt. I think that's how you say that, which is saying that in order for Turing patterns to occur, the inhibitor has to be able to diffuse more readily than the activator. So then this figure is going over the nectar guides again. I should have just pulled up this figure. I forgot that this was here actually. So we are looking specifically at the spotting pattern in the nectar guide. And when we have a mutant which causes the activator to not be present at all, you can imagine that if the activator is what's causing the pigment, the red pigment, if there's no activator, then there won't be any red pigment. In the wild type, we have some amount of Negan, excuse me, which causes these small red dots. And what we believe is that we can tune the effectiveness of this Negan to produce larger spots by changing the situation of how strong the activator it is relative to the inhibitor. And there's a few questions about the different mechanisms for that. Halfway around this pie chart, we see what happens if you have half of the RTO being a knockout. So if you consider a heterozygote where one of the RTOs is functional, the red tongues is functional, whereas the other one is completely non-functional, you have less inhibitor. So if you have less inhibitor, then you're going to get larger red dots because there is more activator relative to inhibitor. If we continue to tune this and we completely knock out the red tongue, there's no inhibitor at all. And so the activator just continues to promote, and it causes just complete red dominance, no yellow at all. And interestingly, in terms of pollinator interactions, these bumblebees love the dark red nectar guides. So we can sort of imagine a situation, and they say almost the same thing in this paper. You can imagine a situation where we might want to take advantage of this to create mutants to encourage pollinator interactions with Mimulus. So this wheel.
Emily?
Yes.
Can I ask a question? This might be a little off topic, but I'm wondering, so if it seems like there'd be selective pressure for the flowers to start to produce, I guess it would be less Negan and more RTO.
Mm-hmm.
Why don't the flowers do that? You know, is there a drawback basically? I'm wondering there isn't there.
Well, for one thing, this red tongue mutant, which creates the completely red nectar guide, is lab produced. So I don't know that it's ever been found in a naturally occurring population. And I don't know selectively if it were to occur, because presumably over evolutionary time, it's occurred somewhere. I don't know what the reason would be why it wouldn't sort of dominate. My hypothesis would be that even though it's attractive to bumblebees, it's potentially unattractive to other pollinators, because there is quite a diversity of Mimulus in terms of their pollinator mechanisms. So what we see is that a lot of species of Mimulus are actually naturally completely able to reproduce, but in the wild they don't cross very often. So they are, the barrier to reproduction is due to the fact that they just don't interact with the same pollinators. So I have no real answer for you, except that would be my suggestion.
Yeah, okay, makes sense.
Yeah, so I would just add, this diagram might be misleading in that it's sort of suggesting that the redder you are, the more attractive you are to a pollinator. And I'm not sure that's what they really mean.
Well, that is what they said in the paper.
Yeah. Pollinators can be attracted to specific flower coloring and patterns, is what's said in the legend. So there are experiments, I think, where they show that, you know, a redder nectar guide might be more attractive to a pollinator, but you know, those are really it's not clear like how similar to an ecological situation that really is.
That's fair.
And the other thing to remember is that flower color is not necessarily only related to attracting pollinators, right? It could be other things that are other reasons for it.
Yeah.
For
Usually
example,
color, pattern, and smell are the three things that people talk about.
yeah, with regards to pollinators, and then, you know, there can be aspects of the petal that are serving as a defense. There can be, I read a paper where the color of the petal was changing in response to heat stress on a particular type of flower. So, you know, it's, I don't know, I just wouldn't overinterpret this idea that it's not like that. You know, presumably the answer to your question, and I guess this is what Emily's saying, is that there's some sort of balancing selection, right?
Yeah.
Some reason why a certain pattern is better.
Yeah, I wasn't sure if it was known, but it seems like it's still an active question.
It's not known to me. That doesn't mean it's not known.
And I
But yeah,
do,
there's sort of.
I just had a brief response in alignment with your answer to Daniel's question. In terms of why certain patterns didn't come to predominate in evolutionary time, it might be because of contextualized within ecology. There might be selective, like Greg was saying, balancing pressures where if there was an overpopulation of a certain plant, maybe that pattern ultimately wasn't really advantageous within the context of the ecological system.
Yeah, and in terms of balancing selection based on what I know about anthocyanin, I think there's a good chance that it either is energetically disadvantageous to produce that much of it because it's costly, and or it causes different interactions with sunlight because it does, it works as a sunlight filter because it's a pigment. So it might cause some problems in that way. Those are sort of my further comments on that. Any other questions? Okay, so if we look at this guy, this is sort of our cartoon representation of what's happening in terms of our red tongue and Negan mutants compared to the wild type. So that original image that I showed you, actually, do we want the full image or do we want the cropped image? We'll start with the cropped image. So if we look at these mutants, well, actually I was saying mutant and then I was hovering over the wild type, excuse me. So we look at the wild type and we see sort of this natural interaction on the nectar guides between the activator and the inhibitor. But if we completely knock out the Negan, then we get just these solid yellow, soft nectar guides with nothing telling the pollinator where to attach. But we still see in the petals that we have anthocyanin, because anthocyanin is that red pigment. So our petals are still pink. They're still having that anthocyanin pigment, which is what's telling us that this Negan is only acting on the nectar guides. By contrast, if you look at this WD-40 knockout, if you completely knock out WD-40 in the plant, you get basically like an albino looking flower where there is no anthocyanin on the nectar guides, but there's also completely white petals because the WD-40 has not been able to interact with the MYB protein that is in the petals. So you get no anthocyanin anywhere in the flower. So that's going back to sort of this trimer interaction. So if we just knock out the Negan, which is the MYB in the nectar guides, then we still get anthocyanin in other parts of the flower. But if we knock out this WD-40, we can't get anthocyanin anywhere. And then by contrast, not by contrast, if we look at the inhibitor, the RTO, we can look at this heterozygote mutant where one of the RTOs is knocked out, or we can look at what happens when both RTOs are knocked out. And what we see is that we get this very dark red color. Now, interestingly, you would expect this sort of in a perfect scenario, if red tongue were the only thing causing the inhibition, you would think that this would become solid red. What you see is that it's not quite solid red. So there is some sort of natural barrier to anthocyanin or Negan, which is causing some sort of patterning as well, which is kind of interesting to think about the physical barriers of the Negan. So sort of going over the wild type versus RTO mutants again, we get this most mild speckled-ing in the wild type. If you knock out one of them, you get some darker speckles. If you knock out both of them, you get this dark red nectar guide. Questions about that before I close the figure?
And stuff like the knockout, the double knockout on C. It also looks like, is it somewhat affecting like the petal coloring too? Like does RTO do more than just inhibit Negan in the guides?
Yes, probably.
Okay.
Yeah, I agree with you that in this picture in particular, or the dissection figures, it does look as though you get much darker petals. Now, in the original photos, you don't necessarily see that effect as much. So it could be a play of the light.
Yeah, I thought maybe it's just a random thing where it just happened to be that way, because it's also like lower on the petals. It looks like this. It's not like a nice gradient, but it's kind of broken. I don't know. It might just be.
Yeah, that
flower.
certainly wasn't the impact on the petals was not a focus of this study.
Yeah, I figured.
So I don't have a strong answer for that, but I do know that RTO is present in the petals and does act as an inhibitor. So I wouldn't be surprised if your observation holds true. Anything else? That was a good observation. Sorry, that was my high school teacher coming out. Good question. And then, so here we get going to Dan's question about what happens in natural populations. So this is kind of an interesting segue. Here we see the interactions between naturally occurring populations. No, that's not what I want. Here we go. So mimulus naturally occur all the way from like the southern end of Mexico up the west coast of North America all the way to Alaska. That's their native region, though they have also naturalized in pretty much all over the world, including in the UK. So these naturally occurring populations, if you look at their interactions with knockouts, you can see sort of the differential effects of what happens when you have RTO knockout applied to different, well, they're not species, but varieties. We get these guys. So interestingly, this one seems to have a much smaller effect on the nectar guides. You still see a different speckleding pattern, but if you compare like this LRD location to the TAR location, I mean, that's a huge difference in terms of what's happening on those nectar guides from knocking out the RTO. So I just thought this was kind of fun to look at and see how the natural populations interact with it. Different wild types interacting with the RTO knockout.
So it's a really common thing to see clinal variation in the alleles of various genes. So that might be the sort of thing that's happening here. The clinal meaning based on longitude, which is that longitude or latitude? Yeah, I don't know.
Latitude, fatitude.
Latitude, fatitude. So based on your longitude, that sort of determines your climate, right? And so you get these variations in gene frequencies as you move along. And that looks, or in variations of morphologies as you move along. So that's really interesting.
And then the only other thing, I mean, this was a pretty short paper and I didn't want to ask you guys to read a whole bunch of mimulus theory. The last little bit of this review makes a point of pointing out like these questions that have now come to fruition due to this study of the activator inhibitor system. And I sort of wanted to highlight them mostly because my research is really closely related to them. And in particular, whoopsies, this question of, do the activator inhibitor proteins freely diffuse between cells? Do they require a transport system like auxin? Or are the different ranges due to the differences in mobility or protein stability? That one in particular, I think is going to be hopefully one that we can shed some light on once we have our mathematical model up and running, because I do think that we're going to be able to point to patterns and whether or not the decay rate, aka the stability of the activator and inhibitor, is what's causing this differential patterning versus the diffusion coefficients, which we also have in our model. So this is a question that we directly can sort of probe with a mathematical model. And I sort of wanted to highlight that. That was all I had on this paper. Now, if you want me to talk about mimulus for longer, I'm happy to. Can do it for a while, but I don't want to go overboard.
You know, the problem with these papers is they really use up a lot of toner.
I liked the length. Obviously, it was like really, it was only like a page and a half, but that can be kind of nice. Like, it's not as helpful, I think, with in a group like this to be given a paper that is clearly about one specific topic, like one specific experiment or so. And the point of handing it out to everybody is for them to kind of get a, introduction to your field. Like I want to kind of know the terms and stuff so that we can kind of talk about it and talk about what you're doing specifically. And if you give us like a, 15-pager steep paper that's on one experiment, then I'm reading the wrong stuff, So it's like, yeah, it was short, but it was also like, I didn't end up reading stuff that I was going to go over my head anyways, because I don't know the, details of plant experiments.
Yeah, my hope in terms of what you guys would get from this was that you would get a little bit of exposure to a reaction diffusion / transcription factor network and sort of why they're interesting and important in the mimulus. That was my hope. So
one thing that I was kind of confused on is why exactly is it important for the inhibitor to be long range? I didn't really understand when they explained it.
Okay, so I did pull up another paper that has some images that are potentially a little bit more useful for that.
Okay.
So I'm going to share that because I did.
Could you
Go ahead.
Could you explain what the difference between a short range and a long range?
So
inhibitor
long
is?
range in this case just means that it diffuses faster
Okay.
or diffuses further. And if you consider like a full 2D scope and you have sort of these different places where this is going to be being produced, then if the inhibitor can diffuse further, then that means that the inhibitor will meet in the middle and produce this region of sort of not pigment, or yellow in our case, the background color. Whereas where the activator is, there will be more activator and you'll get these spots of patterns. And so when we talk about long range, they either diffuse faster or they diffuse further. And that was actually the question that I was raising. It's split between these two columns, which is unfortunate. But are the different ranges due to the mobility or the protein stability? So it either could be due to them diffusing faster, or the activator could be short range in that it decays faster, so it can't diffuse as far away from its source. Does that answer your question, Jordan?
Yes.
Can I share my screen for a second to show you guys
Absolutely.
something? So if this question, the sort of question that you're asking, Jordan, I guess I'm sharing my screen and I want to, not that, I want to show a visualizer. It's a really good question. And one of the things that that we'll do in our class at some point is talk about the way that you can model this type of process using reaction diffusion equations. So there might be a diffusion coefficient here, and then this would be the partial squared x squared of u, and then maybe you would have minus u divided by tau, or maybe you would think of this as minus beta times u. So in a system like this is simpler than what Emily's doing because there's only one species, U, and I don't have U feeding back and activating itself. I only have U potentially diffusing and decaying away. So this is the diffusion part. And this is the decay part. And tau would be a time constant for decay, and beta would be a rate constant for decay. Now what I want to do is I want to show you that if you do the dimensional analysis on this, if U is like, say, a concentration, then there's concentration here and here, right? So that's going to all work out. So that means that this tau must have units of time. That's why we call it a time constant. That's balancing the units of the T over here. Okay. Now this has concentration over length squared diffusion coefficient. So that means that whatever the units of the diffusion coefficient are, whatever the physical dimensions, when you divide it by the length squared, you have to get 1 over time. Does that make sense? Do you see what I did here? I'm forgetting about u because that's already on both sides. And then I'm saying that whatever the diffusion coefficient is here, when I divide by length squared, I better get 1 over time. And so that means that the diffusion coefficient physical dimensions are length squared over time. And so now let's ask your question, which is, what does it mean for something to be long range or short range? Okay, so you're asking, what is it in this equation that determines the length scale? And what I want you to do now is take D and tau. Those are the only two parameters in the problem, right? Sorry, D and tau are the only two parameters. I want you to combine these in some way to get something that has physical dimensions of length. Can you see how you might do that? So we can let Jordan try to figure this out. Anyone who wants to speak up, this might be kind of new for everyone actually too. This is Spencer's class as well.
So you're combining, just to clarify, you're combining D and tau, which are the only two parameters, in order to produce length.
Yeah, so D has physical dimensions of length squared over time, and tau has physical dimensions of time. And those are the only two parameters up here in the model. And you're asking a question about range of action, which is something that has physical dimensions of length, right? So let's see if, is there any way we can combine these two quantities to get something that has physical dimensions of length?
If you were to, I'm not sure if this is correct, but if you were to multiply them, that would cancel out the time, and then take the square root, that would produce length.
Yeah, that's absolutely right. So this has physical dimensions of length. And this quantity is actually what determines the range of action of the inhibitor and the activator. They have, each one has a certain diffusion coefficient, and each one has a certain time constant for decay. And you can get a bigger range of action by having a bigger diffusion coefficient or having a longer time constant for decay, a slower decay rate. So I think that that's just a really, I just wanted to share that because that's a fundamental insight that you can get just by staring at your equation, right? And saying, well, what are the physical dimensions here? And you learn a lot. You learn that there's two ways to change the range of action, not just one, right? Diffusion and time scale of decay. And then you also learn that the range of action actually depends on the square root of that product. So that means if you doubled the diffusion coefficient, or if you quadrupled the diffusion coefficient, you'd only double the range of action, right? So that's another thing that's a little counterintuitive that you learn from doing that.
And it's a sanity check for me just sitting here spouting my intuition.
Mm-hmm. And that's, I mean, this is where the intuition comes from ultimately. You know, you either hear it from people who have thought like this, or you think like this yourself.
So going back to Spencer's question, which was, why do you have to have a long range inhibitor? I think that was your question.
Mm-hmm.
So if we think about if the inhibitor weren't long range and we start with this little bump, because biology has natural variation. So naturally this cell in the middle has more activator. Now, let's go with faster diffusion. We just learned that we can either do faster diffusion or longer time coefficient. Let's just stick with faster diffusion. So it's a race between the activator and the inhibitor. If the activator outraces the inhibitor, what's going to happen? Well, you're going to promote more activator, more activator, more activator, and the inhibitor is never going to be able to catch up. And you would just end up with activator everywhere. But if the inhibitor can sort of down-regulate these things that are further out, then you can get these spots where the activator is high, but the inhibitor is causing these lower spots of activator as well, because the inhibitor has already come over here and said, no, no, we don't want that much activator. That's the story I'm telling. It's not a very mathematical story. Does it make sense?
Yeah, it does. So you kind of get like the inhibitor goes to most of the places and then you get pools of like high concentration of the activator.
That's exactly what happens. And you can get either spots, or you can get like stripes, or you can get wavy stripe type things. Just fun.
Interesting.
That makes sense. I didn't really realize I had that question until Spencer asked it as well. But I guess the way you explained it kind of makes it sound like it's like the negative feedback loop moves out further, and so you can get to differential expression of or differential pigments, versus if it was the other way around, you'd only have a positive feedback loop going everywhere,
Mm-hmm.
and then you'd have a monotone color.
Right. So, oh, Greg's taking over.
Yeah, so this is a great point. So one of the things I wanted to share is, if you think about it that way, the way Emily's discussing, people will often draw something that looks like this, and they call it the Mexican hat interaction function, where this is space, and then this is positive interactions and negative interactions. So if you think of this type of interaction function, you could get this by having a short range positive thing, and then you add on to it a long range negative thing, right? And you get that. And people find in lots of systems that if you have this Mexican hat type of interaction function, you get patterns. So people will refer to this as lateral inhibition. And for the neuroscientists, this is really fundamental, because most sensory systems have an aspect of lateral inhibition at the very beginning of them. Like in the first couple layers of the retina, there's a lateral inhibition, so that the signals from the photoreceptors have this net effect on the output of the retina.
You said sensory, but I heard spencery the first time.
Sensory systems. So that was one thing. And then Emily, here's another type of answer if you want to impress someone that you're going for a job application. The question was, what's the deal about the inhibitor being long range, right? Well, you've got your Jacobian matrix for the linearized system, right? And so this is the FU, FV, GU, GV, U dot equals FUV, V dot equals GUV. We've seen this all before, right? That's the Jacobian of this. And if you were if you know that this is positive because it's an activator activating on itself, and this is negative because it's an inhibitor that it would inhibit itself, right? then you kind of get this sign structure that looks like this. And let's say that we had the remaining signs were like this. That's a little bit of a question, but let's say that was it. Then here, if we knew it was stable, we would know that the sum of these terms was negative, right? Because the trace of this matrix would have to be negative. But the determinant would have to be positive. This times this would be a negative number, minus this times this. That would be a positive contribution. So there must be something where, well, when you put the diffusion in, where does it go? I don't know, do people understand that the diffusion term ends up contributing something that looks like k squared du up here? So if you look at this, it's like, well, when the diffusion coefficient's added, what you're effectively doing is lowering this entry and lowering this entry.
Oh.
So oh, I'm getting stuck. This is going to be so impressive. So if you look at Murray's 2003 paper, let me grab this.
I thought that was are you talking, does he have a paper as well, or just that textbook chapter?
Just the textbook chapter is what I'm looking at.
Okay.
So let's see if I can find this. So I'm clearly
Like
just
my boss.
I'm clearly just making some great mistake here. So the point is that when you go through all of this analysis, there's the lateral inhibition thing. That's the example from retinal ganglion cells. Here's the Jacobian that we're talking about. Here's the statement that when there's no diffusion, we're supposed to have a stable steady state. So the trace of this Jacobian is negative and the determinant is positive. And then you assume that the system is linearized. W is the spatial and temporal perturbation. You derive an expression that that perturbation solves. Blah, blah, blah. You find that basically you keep going through your analysis and eventually you know, eventually he's deciding that this ratio of diffusion coefficients has to be bigger than 1. He's basically saying this can only happen if the inhibitor is diffusing faster than the activator. So if you want the real answer to the question of like, why does this happen? Why do you get the Turing instability when the inhibitor is faster than the activator? You get that. You get the actual answer by saying, I've got this two-dimensional system, I linearize it, it's supposed to be stable in the absence of diffusion, that gives me a bunch of criteria, right? And now I add the diffusion in, linearize that partial differential equation, I get another Jacobian that now has the diffusion coefficients in it. That one's supposed to be unstable. And you compare the stable and the unstable case, and you see that this can only happen, you can only have the one with diffusion being unstable while the one without diffusion is stable. That's the Turing instability, right? Diffusion leading to the instability. And you see in the analysis that that can only happen if the inhibitors diffuse a bigger diffusion constant than the activator.
Okay.
And that would be, you know, that would be like in a job talk or something. If you could whip that out of your memory bank, right? Even if you can't go through every step, if you could just go through the logic of the argument, it would be really impressive, right? And you can see that I was not able to do it because I was making my little argument here, and you know, some sign was wrong. But this sort of thing, this sort of argument, is the sort of thing that Emily's trying to do in her in a more complicated model where there's an activator and inhibitor from both of the genomes, right? From the mom and from the dad, and that they can have different properties. So it's a really relevant question, Jordan, that you're asking. I guess one difference is that in Emily's problem, it's not the diffusion coefficients that are changing that induces the instability. It's something about the fact that you've got the two different activator and inhibitors, right? So in the homozygous activator inhibitor system for cupreus, there's no pattern. In the homozygous activator inhibitor for variegatus, there's no pattern. But in the F1, where they're heterozygous, there's a pattern. So it isn't the diffusion coefficients that led to the instability. It's something about the interaction between the two genomes, meaning the way that the transcription factors influence the regulatory regions on the opposing gene.
I still think that you have to, we still need to debate this, but I still think that you have to classify it as a diffusion-driven instability for it to be a Turing pattern. Even though we're not changing the diffusion coefficients, we still need to check that that's what's leading to the instability.
Yeah, I think you'd probably be right about that. I mean, we wouldn't want to call it a Turing instability unless that was happening. But in a way,
I think we still have to have that sort of dichotomy of stable without diffusion, unstable with diffusion. It's just that we're not changing the diffusion. We just have to check that condition.
Yeah. And I don't think we need to insist that the parents cannot go unstable ever, even if the diffusion coefficients are different.
I think we proved that there's no situation in which they can't go unstable given sufficient diffusion.
Yeah, I think that's right. In a way, I feel like that this observation that you just made is good because it gives us more conditions, right? The more we can say about the system's properties, then the more chance we'll be able to say something analytically. Anyway, does anyone have anything else to say or ask of Emily? I felt like that was a really nice presentation, and it was, I feel, helpful to have just a real short paper and an overview.
Yeah, I think if anyone else decides they want to do an overview, I recommend it, but I also recommend that you have some images ready for questions that you think might happen, because in an overview, you really have like maybe one figure. So
yeah,
that's my recommendation.
And when you do this kind of presentation, Emily, some questions occurred to me. I'm wondering if you know the answer to this. So you described red tongue as being self-inhibitory.
Yes.
So is red tongue actually a MYB type transcription factor?
I don't remember.
Okay, so I think I know the answer, but I'd like you to look it up, okay? So, because I could be wrong. So the question is, red tongue self-inhibitory, but is it self-inhibitory because it's sopping up the beta helix loop helix component of the Negan trimer, and Negan activates red tongue, so that has the effect of being self-inhibitory? Or is it self-inhibitory because red tongue actually is a transcription factor itself? And then one of my questions here was, why does the inhibitor have to be long range? Jordan, so good question, Jordan and Spencer. The other question in my mind is, when you read about the MYB transcription factors, you'll often read the R2, R3 type MYB transcription factor, and that's, Negan is one of those. But then there'll also be like R2 transcription factors, R3, R1, R4, whatever. So we should probably know just a little bit about the molecular biology of like what that nomenclature means. I think the R.
Yeah, I had that under my belt when I did the prospectus. I read some papers on the molecular biology of that. And I can pass those papers along. They're dense.
Yeah. And yeah, so we don't want to get too lost in it, but
Right.
I think it stands for repeat. And if there's obvious functional differences, depending on whether you're an R2, R3, or just an R2 or just an R3, then we should probably know what that is, because, you know, maybe RTO is one of those, and we ought to know that because that'll tell us kind of something about how it's operating. Another thing that's even more basic is if you were at a job talk or something, and somebody said, well, why is this called MYB? Any idea what the MYB stands for?
I just tried Googling it during our thing, and I couldn't find it. Like, it wasn't, Wikipedia didn't have it as like its first thing.
I don't remember.
I think it's standing for myoblastoma.
Is it?
Which is a cancer. You know, I think that means a muscle cancer, right? So one of the things to remember is that a lot of these transcription factors were first found in some context where there was abnormal cell growth, right? And so you could have a mutant in some transcription factor leading to cancer. And then as people do the research, they find this particular type of transcription factor that's relevant. And then you compare sequences with other sequences, and you find, oh, wow, there's this whole family of transcription factors that have these properties. And so I think that's how these names kind of get propagated. And so you'll have something that's referring to cancer in animals when we're talking about just normal development of flowers. But
it is myeloblastosis virus that was found originally in the avian myeloblastosis virus.
Myeloblastosis virus?
Myeloblastosis virus, yeah.
Wow, okay, so I was wrong. It has nothing to do with it. I thought it was a cancer. Anyway, so just having a little bit of that history under your belt might be nice, because it could come up when someone asks some question. It's always good to kind of know that sort of thing.
It is an oncogene.
Okay. So I guess this is one of those ones where there's a virus implicated in the cancer.
Yeah, I don't know. I would have to go look it up, but it's apparently in birds.
Looks like the virus can cause sarcomas and other tumors.
So Dan, I found this nice little. It's a little bit longer than Emily's, but it's from the same journal, Current Biology. It's one of my favorites. And it's a primer on the importance of adaptation in neural network function. Do you think that it's 6 pages. And there's like 4 or 5 figures, like, you know, explaining an adaptation scenario. But would you be willing to maybe go through this for one of our meetings next week?
Yeah, for sure.
Okay.
I could do Tuesday.
You can do Tuesday? Okay, I'll send this to you and everyone else. And then
cool.
Emily, this one, have you read this?
Yes.
this paper is kind of an overview of why monkey flowers, why Mimulus is a good system to work in. So that was really interesting. I'm going to send this around to everyone just because it was one of the papers cited here that I feel is helpful if you want to read a little bit more. And then there's also a couple other papers that I'm not sure, Emily, you've read yet that I'm going to send around. Like there's one here about
I think having read that paper and listened to the Heredity podcast, I think that they talk about largely the same things. If you're into podcasts, the Heredity podcast is, I mean, it tells you basically the same things in an interview format.
that's nice.
I can send that around. Greg found that one.
Some of your, like you had a paper that you showed us about the role of auxin transport and plant patterning.
Yeah, I found that one because it was linked. I actually haven't read it very closely yet, so I didn't want to go too deep into it.
So, but this is one of the things, just as you guys do your research, finding like a entry level paper like this, right? And then you look at the references and you can find like 10 nice things that these guys are basically saying, these are the papers that sort of frame this whole problem. And so you would want papers like this to be in your prospectus and ultimately in your dissertation. So one of the things I really would recommend you all do is have maybe on Overleaf or on Zotero or something like that, have like the what I would call an annotated bibliography. So where you have like the paper and then what you thought about it, but then there's actually the bib file or whatever you would need in order to put that in as a reference in a paper that you write. Because then you sort of have your, you kind of have your library of things that you've read and are familiar with that you're very likely to reference when you write your first paper. Right. So it's a really good thing to do. I
was a big fan of having like physical note cards when I was writing my prospectus. But I do think that in terms of longevity, having it digitally in Zotero would have been good.
Yeah, for me, I go back and I kind of do both electronic and physical. For some reason, I really, really appreciate having the physical copy of this thing, paper, as I read and I underline and stuff. But then I also have the electronic version of the paper someplace where it's sort of indexed in a way where if I just searched on it, like, do I have this paper already? You know, it pops up in Evernote. And then I have a couple different .bib files that have, you know, the bibliography information for papers. There's one that's called math. There's another one that's called metabotryptoglutamate receptors and so on, right? And I try to keep those somewhat up to date. The truth is they end up getting expanded every time I write a paper. But if you have, especially for you guys at the stage that you're at, you can easily have every important paper like printed out in a stack somewhere near your desk, right? And having it like that and being able to flip through and remind yourself of things can be really, really helpful. I mean, it's important to let's see. One of the principles of how to be a good researcher is make sure that some good fraction of every day you're being a scholar, or maybe for an experimentalist, you might have to say every week because sometimes the experiments take the whole day, right? But make sure that some fraction of your time you're doing scholarship. That means you're reading papers, you're writing in your notebook, thinking about why you're doing what you're doing and what you should do next, right? And, you know, you'll be amazed at like how that can get you through a depression. if you're feeling like, oh, this is boring, why am I doing this, you know, blah blah blah. If you start reading the papers, you'll remember why you're doing it, right? And it's really important because people will, you'll find the more you read the literature that there's people out there that are just doing crap and they're just trying to publish it as fast as they can. And you don't want to be one of those people. You want to be someone who actually knows the literature and does something that's a contribution to the literature, right? You don't want to just be operating on your own, pretending that you're the only one that's ever thought about this stuff. I mean, it's completely counterproductive, and it's not even true, right? It's not the way science works. So I really, I think that anyway, I find it's almost like once a month, I'll be in a slump and I'll realize, oh, it's because I haven't been reading enough papers. That's why I've lost track of what I'm doing here. Didn't they just start reading papers or watching a YouTube video where somebody's giving a lecture on flower anatomy or whatever, right? And it's like, oh, that's why we're interested in all this.
Yeah, I do the YouTube lectures. I have like a few saved full classes that I found that I'm like, oh, I would like to learn about this. And that's my like, okay, I just need to like learn something and stop banging my head against this code. That's where I go.
Yeah. And the whole thing, the issue of like part of having a team, right, is that we learn from each other. So, maybe, maybe Spencer's the person you'd ask a graph theory question of. Emily's been doing a lot on reaction diffusion equations, maybe, so kind of knowing like who you can talk to get a question answered is really good. And then what we're doing here, reading a paper together, is I think just really, really, really key. Okay, so our next meeting is do we have a Whitman meeting at 1 o'clock, Emily? We do? Okay.
We do.
And then we have a UPSI meeting at 9, right? Tomorrow?
Mm-hmm. And then no biomath.
No biomath this week. Class on Monday and group meeting on Tuesday, and that's when you'll be talking about this paper that I'll share, right? Okay.
Yeah.
All right.
We're just doing the paper then, not my like research, more like just focusing on almost like fundamentals of the neural oscillations. Or do you want me to try and apply it to what we're doing?
Yeah. I mean, you can read it and then decide, okay, of these 3 different types of adaptation they're talking about, is really this one that's the most relevant to me, and then explain why or something like that.
Okay.
Do whatever you want with it. I mean, the criterion is design it so that the prep you do is good for you, right?
Mm-hmm.
And so that it's what you ultimately present is interesting for everyone, or useful for everyone. That's the way I'm thinking about biomath like that right now too. It's like, okay, I got to do something here. And so I'm going to be spending, 2 days worth of work preparing this biomath talk. So why don't I make sure I pick something where the work that goes into that is actually something that I needed to learn or needed to consolidate. So even if I don't reuse the slides or something, that maybe conceptually I needed to consolidate the information.
Yeah.
Gordon, you're looking sleepy. Are you sleepy?
You're muted.
No.
There you go.
Oh, were you talking to me?
Yeah. I just saw you. I saw your eyes.
Oh no. Sorry.
I think you were probably reading something on your desk or something.
Yeah, no, sorry. My internet connection has also been very unstable this entire meeting, so
Yeah.
I'm just,
Hanging in there.
waiting. Yeah.
All right. You also, I'd like you to give one of these presentations down the road too. And so it'll be, it's not a big deal because it's just us, right? But it might be good to practice doing something like this before you do biomath. So if we can find some little paper, just a couple pages that's relevant to your project and share it, that would be good. All right, see y'all.
I'd really like to hear about, you do like studies of big genome analysis, right?
That's probably something that I'm going to do right now. focused a little bit more on phage research because I'm helping with a phage project. But yeah,
Okay.
it's something.
Because so selfishly, the big genome analysis stuff, like I don't understand it. And I feel like I should understand it for the genetic research. So would love for that kind of a talk, if I can put my 2 cents in.
Yeah, like maybe some kind of overview of some, what would we call it? Genome science method.
Like, what are the modern techniques? What are their shortcomings? Mathematically, how rigorous are they? Like, these are my big questions where I'm like, I don't know how much of this is stats nonsense and how much of it is like actually valid.
Mm-hmm.
I have issues.
Yeah, I can try to find maybe an introductory kind of a paper.
That'd be great.
Yeah.
Got a lot on your plate though, don't you?
Yeah.
Okay. All right. Well, everyone have a good rest of your day, and I'll see Emily in a couple hours. Bye.
See you guys.